Hello students, welcome to Vedantu NEET English. I am Ms. Gopika, your own biology master teacher. I hope I am audible, visible and the presentation right behind me is also visible. Please let me know in the chats if we are live yet. Yes students, tell me if we are live and I am clearly visible, audible and everything is going proper. Yes, I think we are live, right? Very nice, very nice. Okay, I can see a lot of energy. So happy for all of you who put a lot of fire emojis in the chat before the class. Okay, so what are we going to do? Today we are going to do important concepts of human health and diseases, and as you know, human health and diseases is a very long chapter, right? If we study at one shot, it might take us 5 hours to 6 hours to properly understand everything and then study it. So what I have been doing is I have been picking up some important concepts from where I have seen a lot of repeated questions. Okay, yes, very nice. Okay, so this way what happens is you do a revision of it, but because since we already completed our zoology portions and it is very fortunate that we have enough time for revisions as well, right? All of you quickly mark your attendance. How do you mark your attendance? By clicking on the like and also sharing it with your friends on WhatsApp and Telegram. Okay, so shall we start? Very nice. Okay, 1 second, I think there is one. Where did that person go, students? Yeah. Okay, done. Okay, done. Okay, all of you like the video quickly and then we will start the class. Okay, so as last class itself, I have brought blank slides and I am going to teach you and we are going to write. So you can also make short notes with it. So don't wait for until I share the notes. Make kuti notes. Okay, next to your NCERT itself, write few information. So what are we going to start? Last class we understood about the immune system, how our body is going to work, and when an antigen is going to come, how our body is going to react to it, and with the help of two types of immunity, humoral immunity and cell mediated immunity, we understood that our body's immunity works in such a way that it's my ability. It's my body's ability to do a task, right? Ability to fight a pathogen, fight anything, that any foreign particle that is coming from outside. That is why during graft, okay, that is transplantation, graft rejection happens, because when you are taking something from someone else's body, it is understood that it is a foreign particle. So to suppress all of that, we use immunosuppressants. All this we studied in last class. So shall we directly dive into this class? So today we are going to study about AIDS. First, I want all of you to tell me the full form of AIDS. Come on. First question to all of you. Tell me the full form of AIDS. Quickly in the chats. Okay. Tell me the full form of AIDS. Hello, hello, hi. Hi Madhumati, hi Jerusha, hi Madre, Sukanya. Okay. Volume is not high. Volume is high only, no? Okay, volume is fine, right? My voice is not clear. One second, let me check. Voice is fine, no students? Here audio is completely high. I don't think more than this the volume will be increased. 1 second. Yeah, everything is at the maximum. I think it's because of the, once I start writing it should be fine. Okay, once I start writing it will be fine. Clear? Volume is too low. Should I call them? Every day one problem will be there. No students? Now it's become a regular thing to have this many problems in life. I think just adjust for now. I will just inform him. Okay? You people are not wanting the class that much, I guess. That's why every day there's one problem. Yes, 1 second, I have informed. Okay, let him come. Till that we will continue our class. Okay, we'll continue our class. Okay, very good. Acquired immunodeficiency syndrome, right? That is the full form. Acquired immunodeficiency. Yes, they're telling the volume is low, but if you hear in my phone, see, this is the volume. It's actually low. Is it the OBS? Guys, 1 second. some bus sound is coming up. One second. Okay. Is it better now? Hello? Is it? Okay. Just 1 second, okay? Is it better now? Okay, yeah, it's the battery's problem. They'll fix it, okay? Yes. 2 minutes, we'll start the class, okay? I'm so sorry. I don't know why this happened. Yes, understood. The bus sound got increased, no? Yeah, they'll fix it. 1 second. Hello students, is it better now or is it fixed completely? Yes, fixed. Tell me quickly. It's 20 now. I think. Tell fast. Is it better now? Clear? Okay. Thank you. Okay, done. Okay. Yeah, she went. Right? Every day she is there. She curse has come, no? Because you people did not join the batch. Yes, fixed. Okay. Did we make the likes? That you people didn't do. Quickly mark your attendance, all of you, and we are going to start the class. Okay, starting one problem is there. It's good students. Nazar will go. Okay, so acquired immunodeficiency syndrome. Okay, This is the full form of it. Now ma'am, what is this? See, acquired meaning what? That it is not happening, but see, it's not necessary also, right? Because sometimes it can come from the mother to the child, okay? So that can happen. But otherwise, if you see, acquired immunodeficiency syndrome, from the terminologies, we understand that it causes deficiency. That means it affects your immune system, right? It affects your immune system and it is going to decrease your immunity, right? Your immunity will get decreased if you have AIDS or if you are tested AIDS positive. Now syndrome means what, students? All of you, last time also I had told this to all of you. What is the meaning of syndrome? Syndrome means a set, like many things coming together, right? Many things coming together means what, ma'am? That means many symptoms we see, right? Many symptoms when it come together, we can call it a syndrome, okay? Many symptoms when it comes together, we can call it a syndrome. Clear to all of you? So I have decoded the word for you. Now let's see the history of AIDS, like how it was first understood. So students, AIDS were actually first called as SIV, but I'm not going to go into the depth of it. So the first time any person, like let's say when they first notified or was first they got to know about AIDS, was from gay community, okay? That means homosexual people, right? Gay community, it was first ever found out. Gay community, okay? And because it came from the gay community, people thought it had something to do with the sexuality. Now imagine the first ever case of AIDS came from a gay community. And maybe the second case also came from the gay community. Now they will be like, okay, wait, it has something to do, or it might be something very related to the gay community. Understood? So this was the first findings or first thought process was that, okay, it is because of the sexuality. Later in different parts, especially in India also, in Chennai or something, the second case appeared and it was in a woman. So they understood that, okay, it has nothing to do with the sexuality. Okay, I want all of you to listen to me. Okay, it has nothing to do with the sexuality. It is a immunodeficiency syndrome. Clear to all of you? It was also called as GRID, okay, because again, gay community based. So it had nothing to do with the gay community, and but the first case has been come from this same community, okay? Now later they understood that it is not something to do with this, but it can happen to anyone, and there is a virus that is affecting this, which is called as your HIV. What is the virus called as? HIV, with the full form. What is the full form of it? Human immunodeficiency virus. Clear to all of you? Yes or no? This was the second thing. Clear? All of you, no doubts, right? Very nice, very nice. So the discovery happened from the gay community. Later that thought process went that, okay, it's not because of any sexuality, it is happening to everyone, okay? And then they noticed that the virus that was causing it was called as HIV virus or human immunodeficiency virus. Now the thing about this virus is, students, this virus is a retrovirus. This virus is what virus? Retrovirus. That means what? Because we found it in the 19th, so we call it retrovirus. No ma'am, we call it retrovirus. Why? Because normally according to our central dogma, now what is central dogma? All of you learned molecular basis of inheritance, right? DNA becomes RNA, RNA becomes protein. That is the central dogma of your molecular biology. Now normally that is what is supposed to happen. That means from, let's say, from DNA, RNA should be formed. From RNA, protein should be formed. Now you tell me, what would be reverse transcription or retrovirus? What are they going to do? They are going to do something called as RNA to DNA they will be able to form. So this happens with the help of an enzyme called reverse transcriptase. Students, this is just the introduction, okay? This is just the introduction. Reverse transcriptase. So retrovirus means the virus that has the ability to form from their viral RNA, they can form viral DNA. They can form viral DNA. Is this clear? With the enzyme called reverse transcriptase. Okay, with the enzyme called reverse transcriptase. Is this clear to all of you? So far good? Yes, easy peasy, easy. Okay, so it is called as reverse or retrovirus. Okay, and the enzyme they have is reverse transcriptase. Now, shall I ask you all a question? Now, let's say one person. Okay, this is that one person. This one person is tested HIV positive. This person is tested HIV positive. Does that mean that this person has AIDS? Does that mean this person has AIDS? Does this person have AIDS or not? Yes or no question. All of you can tell me your input according to that. Okay, anything you want to tell, you can tell. Okay, yes or no question it is. You have to tell me yes or no. Yes, quickly tell me. Wow, wow. Yeah, exactly. Albert Einstein, Newton, okay, Tesla, all the people are here. Yes, tell me. Quickly answer this. Okay. Madhumati is trying to get the attendance done, but these people are not doing. Okay. Yes, guys, tell me fast. I'm waiting for your response. Tell me, if a person is tested HIV positive, does that mean that person has AIDS? The answer for this is no. You will be shocked. What? No. No, it's not necessary, because HIV positive means during the test we understood that this person's body, the entry of HIV has happened. But what is AIDS? AIDS is when the HIV virus continuously starts killing your T helper cells. Okay, that means it basically, let's not even get into T helper cell. Let's say it completely reduces your immune system and opportunistic diseases start coming. That means a normal flu, a fungal disease, let's say your TB, okay, tuberculosis, all these diseases starts coming. Okay, that is when I tell that this person has AIDS. That means HIV has to keep increasing in the body, it will go to a chronic stage, then it will go to a deadly stage. Okay, see, the death is for sure. If a person has AIDS, it is possible, but we can increase the lifespan. Now students, just because a person is HIV positive does not mean that person immediately has AIDS. The AIDS will be happening maybe after 5 years, maybe after 10 years, maybe after 20 years. Only we can tell that this person is AIDS positive. Okay, completely has AIDS. Is this clear or not? Is this clear? So you understood the concept. So HIV positive can happen to anyone. That is, human immunodeficiency virus can be there in your body, but when it multiplies and continuously forms this virus, right, and it goes through a chronic phase and then it goes to AIDS. That means even a small fungal disease, even a small disease, okay, a bacterial infection can become deadly in that person. That is when we tell that this person has AIDS, because the complete immunity has been reduced. Clear? Done. So this is your introduction to your AIDS. Okay, so retrovirus I have explained. Okay, some places you will see lentivirus also. Now as of now, don't break your head, just understand retrovirus, family of that, many things are there. Okay, so this is your introduction to it. Now you understood that a person who is, I can also make a statement telling that a person who is AIDS positive, that is a person who is tested positive, okay, who is showing positive in AIDS, or let's say a person who has AIDS is HIV positive. Okay, is HIV positive, because obvious HIV has increased, that's why this person has reached to an AIDS. Okay, clear to all of you? Yes. So this is your introduction to AIDS. Now I will teach you about the virus, okay, because you have to learn a lot about the virus. So students, introduction was clear to all of you? Yes or no in the chat box? All of you tell me. Clear? Enzyme is clear? Okay, very nice, very nice. Enzyme is clear, introduction also is clear. This is a immunodeficiency virus because it is going to reduce the immunity. Now shall we draw the virus? Okay, shall we draw the virus? I did not keep an image. Okay, I thought I will draw it right in front of all of you. Okay, so you will also be able to understand, okay, this is how the virus looks. Okay, now this is the virus. Yes. Okay. Now all of you, if you want, you can draw with me because there are some important labelings that you're supposed to know. Now students, look here. This is your layer which I have drawn. Okay, this is one circle I have drawn. Now what is this circle? It is your phospholipid envelope. Okay, it is HIV is enclosed in a phospholipid envelope. Now on this they have something called as the docking proteins, okay, which are called as glycoproteins. So first I am going to draw that. So if you see like this, okay, it looks like one lollipop kind of structure like this. Docking proteins are there, or we call glycoprotein. Each of the glycoprotein has a name. Okay, so can you see this? I'm not going to draw the full. Can you see like this? Glycoproteins will be there. I'll just draw few of it so that I will be able to make you understand the concept. Okay, this is clear. One side I have drawn. Now students, in this, each of it has a name. Each of it has a name. Matlab what? This circle has a different name. This one has a different name. Now remember that these are glycoproteins. What are these? They are glycoproteins. They are glycoproteins. Now as I told you that everything has its own name. So this one, okay, this one which you're seeing, the circular structure, this is called GP, okay, glycoprotein 120. This you have to know. It is necessary for you to know. Okay, what is it? Yeah, like Deekshi sent emoji, you know, same way. Yes. So here what is going to happen, students? This circle is called as GP120. Why is this important for you to remember? Because this GP120 is going to be the one who is going to bind with your CD4 receptors. Whichever cells in our body has CD4 receptors, this HIV's GP120, glycoprotein 120 can go and bind. Okay, that we will go into, but right now try to understand the structure. Now this one, what is this? Can anyone tell me how much it is GP? All of you open your textbook and notebook and see quickly in the chat. Tell me, no one is there to tell? Something and all you people are talking, yeah? I can't understand only. Okay? Now come back to the class. It's a very, very important concept. You know how many questions come? And students, end of the session, I have added questions also. So stay till the end of the session. Don't go anywhere. Answer the question, then only go everywhere. Okay? Otherwise, you will not know how much you understood from the session. And if you do not listen properly, that's not my mistake. Yes. So, I have done the whole thing. Okay. Now, it looks very nice, no? Yes. Very no. Good. GP, who told that? GP41. Very nice, Sony. Very nice, Sukanya, okay, and Madres. GP14, ma'am. This line structure which I am seeing is GP41, okay, GP41. Now these are what? Glycoproteins. Clear to all of you? Now, when we go inside the structure, okay, this is the phospholipid layer, okay, phospholipid envelope is there. I'm just writing the key points here. Phospholipid envelope. Okay, clear? Phospholipid envelope is there. On that we have docking proteins, or also you can call, okay, docking glycoproteins I am writing, so you remember, okay, glycoproteins. Fine, clear? Important points are in one side. Now inside that if I go, I'll be able to see a capsid. Okay, I'll be able to see what? A capsid. So let's draw a capsid, and a capsid is usually circular. So on this structure only, I will just draw the circles. Okay, so that drawing also will be easy for me. Okay, so 1 second, students. Yes, so this is your capsid layer. All these notes will be available on Vedantu NEET English Telegram as well as Biology by Gopika. So those who have joined new, don't think that, oh my god, where will I get all of these notes? These notes will be available on the Telegram. Okay, students, what have I drawn in the between? This is your capsid layer. Okay, this is your capsid layer, and I have taught you capsid in biological classification. Yes or no? The protein coat, okay, made up of multiple capsomeres. So this capsid is made up of multiple capsomeres. And this is a protein code. Inside the protein code, what will be there in virus? What will be there in any virus? Doesn't matter, retrovirus, COVID virus, that virus, this virus, not retrovirus, HIV virus or coronavirus. What is going to be there inside that? Your genetic material is going to be there. So here they are going to have two double-stranded RNA structures. What are they going to have? Two double-stranded RNA structures. So these are your two double-stranded RNA structure. Clear to all of you? Yes or no? Easy peasy? So this is why I drew it in the class, so that every step you will understand. Okay, every step you will understand. This is going to be your single strand, sorry, did I say double stranded? I'm so sorry. Single stranded RNA structures. Yes? Done? Done and dusted? Any doubts in this so far? Okay, very nice, very nice. All of you are understanding, right? Okay, single stranded, yes, yes, single stranded RNA structures are there. Now, inside that, students, a very, very important thing is going to be your reverse transcriptase, right? We already spoke about it, so there's nothing to worry about it. We already know that there is going to be reverse transcriptase, which are the enzymes present. Okay, enzymes present is called as reverse transcriptase, which is going to make the RNA. This is RNA, no? Now, if it has to enter my body, I'm the host. If it has to enter my body, mine is a DNA. So, it will make sure that RNA to DNA will be done with the help of reverse transcription. So, here, this is going to be called reverse transcriptase. Many students will have a doubt that, ma'am, is enzyme present? Yes, enzymes are present, okay? 3 enzymes are present. Some people teach you that, some people don't, but yeah, reverse transcriptase is good to know, okay, no harm in knowing. Now, then you have integrase, okay, then we have an enzyme called integrase, which I am drawing it here, okay, this is called as integrase. Why is this integrase there? So that we can make sure that this viral DNA gets into the genome and, you know, we integrate, okay, we integrate, got it? Yes, Tanuja, I can see your message, don't worry. Okay, I can see your message. You be in the class, that is more important, no, than your messages being there. You should all be present in the class, okay, because I am doing a revision series, and if you people are not benefited out of it, then there is no use of doing it. Okay, so integrase, I have drawn it in blue color. Got it? Now, students, we have one more enzyme which is called as protease. One more enzyme which is called as protease. Okay, so like that we have come to understanding. Where do I draw it? Yeah, okay. Come to understanding of what is the structure of your HIV. Okay, HIV. So, the enzymes I am going to underline it with yellow. Okay, protease, integrase and reverse transcriptase. Okay, reverse transcriptase. Clear to all of you? Can we go through the structure once again? So, listen to me carefully. This is the phospholipid envelope and this is how your human immunodeficiency virus looks like. Okay, this is your entire envelope. On the envelope, you can see docking glycoproteins, or simply you can just remember glycoproteins. Okay, now, these glycoproteins, you will see that they have a circular structure. 2 proteins are there, no? So, this glycoprotein is called as GP120, which is going to go and bind with the CD4 receptor. Okay, which is going to bind with the CD4 receptor. Clear? Now, down to that we have GP41. Okay, GP41, that is going to, yes, capsid I have labeled here, protein code is labeled here. What happened, Sony? I did not understand your message. Protein code, ma'am. Yeah, protein code is labeled. You want to know the name. Okay, the matrix. That is your P17. Okay, there is one matrix above this, but it's okay if you don't know that also. Okay, there is one small matrix. The same protein code only we tell. Okay, which is your P17. Okay, P17. Clear? Is that the question, or did I not think of your question? Okay, fine. Answer quickly. I have to wait for you people. There's a matrix in the outside, students, which is your P17. P17. Okay, it's the matrix which is just quoting the protein code. Clear? Yes? Okay, fine. That only you asked. Okay, clear? Now, students, look here. This protein in that GP120 is going to bind with the CD4 receptor. Now, ma'am, what is inside them? So, then as Sonia asked now, there is a matrix which is called as P17. Okay, the protein code is P17. Inside that, there is a capsid layer which is going to protect the genetic material. Normally, in every virus, it's the same. There's a protein code, inside the genetic material is there. Here also the same thing. This capsid will be there. So, students, when it is infecting the host cell, this capsid with the you know, genetic material and the proteins is going, sorry, enzymes is going to enter inside. Clear? So remember like this, when it is going to infect the cell, it is going to enter inside the cell with the entire capsid. Okay, the glycoproteins and the everything will be outside. So this whole thing will enter inside the host cell. So now look here students, after that you have a single stranded 2 RNA strands. On that you have your reverse transcriptase, which is going to make sure that the conversion happens from RNA to DNA, and then you have protease, which is also going to help in the formation of the viral protein. Then we have integrase. Okay, clear? To do the integration. All this makes your entire human immunodeficiency virus. Now the most important part is that, ma'am, how do this HIV exactly enter into the host cell? That question can come to all of you, no? Ma'am, how is this happening? So students, remember that HIV will always, or especially your GP120, will go and exactly bind to a receptor which is called as CD4 receptor. Okay, so let's say if this is your, okay, this is your T cell. Okay, this is your T cell. On the T cell there is a receptor called as CD4 receptor. Okay, this is your CD4 receptor. Now what is the full form of CD4? Cluster of differentiation. Okay, this to the CD4 receptor, exactly this fellow, who is this fellow? The GP120 will come and bind like this. Okay, it will come and bind like this. Clear to all of you? Yes or no? Fine. Now, which are the cells who have CD4 receptor should be your next question in your head. Ma'am, which are the cells who have CD4 receptor? That is, students, this CD4 receptor is present in cells like, okay, present in cells like your dendritic cells, okay, macrophages and your T helper cells. Okay, T helper cells. Now in this, one very important thing, that question is going to pakka come. Okay, GM artist, yeah. CD4 is present on the plasma membrane. Okay, here it will be present. So now this is the structure on the cell. Like this, it will be present. Clear? It will be present on the surface like receptors, where it will be present on the membrane, right? On the membrane it will be present. GP120 will go and attack. Clear? Yes. Dendritic cells. Thank you so much, Mani. Thank you so much, Deekshi. So students, this on the dendritic cells, on the macrophages, on the T helper cells, the CD4 receptor is there. But the scariest part here is what you know, that this HIV is going to consider macrophages as the factory, okay, as the factory of forming. Why are they considering as a factory? Why am I telling that they are factory? Because they are going to form their new viruses in the macrophages. Now ma'am, what about T helper cells? T helper cells, the problem is, if the HIV affects the T helper cells, they are going to directly harm the T helper cells or kill the T helper cells, so that your immunity is going to decrease. Clear to all of you? They are going to kill them, right? Kill the T helper cells. So this is the difference, and the question is going to come on this, which is the factory of HIV. Which is the factory of HIV. Any doubt so far? Yes, very good. Macrophages are the factory. Clear to all of you? No doubts, right? CD4 receptor, your CD4 means cluster of differentiation, okay? Cluster of differentiation. Cluster, no space to write only. 1 second. Cluster of differentiation. Clear? Yes or no? Super duper clear? So far good? No doubts? Very nice, very nice. So we'll continue, okay? So this is your HIV virus, done and dusted. Now, students, don't get confused, okay? See, orange and yellow is very clear in the photo. I mean, when I send you the PDF, here it will not be clear, but don't get it confused. This is the outside envelope. This is your capsid layers, okay? Now, exactly what is going to happen? One second. See here, I have just added an image so that you can understand that the diagram I drew and the image, okay. So this is how your lipid layer is going to be. See, this is your phospholipid envelope, this is your docking proteins. You can see that this is your GP41, okay, and this is your GP120. Clear? This is your GP120, and you can see how the protein coat is, right? Protein coat is like this. Now, if you open that, what do you see? You will see that there is a, again, glycoproteins are mentioned. There is a matrix core which was asked by Sony. This is the matrix core which is P17, and then you have a capsid which is P24. If you can remember, otherwise it's fine, because in NCERT nowhere it is mentioned, okay. Two codes are there, one matrix is there which is P17, and inside the capsid is made up of P24, okay. Then students, again, nucleocapsids are there, don't need to worry about it. Now, as you go inside, see, this is the capsid membrane. Inside you will see the single-stranded RNA, you will see the reverse transcriptase, you will see the protease, you will see the integrase. Clear? Yes or no? And then the lipid membrane is there. So this is the structure, exact structure I have drawn, okay. Yes, you should study it, Monix. Okay, because everyone believes that whenever an HIV structure is taught, minimum they will teach about, okay, GP120 and GP41, okay, because this one, if you do not know this, you will not be able to understand who is going and attaching to the CD4 receptor. So it is necessary for you to know this. Even though it is not there in your NCERT, it's okay. Teacher's responsibility to put it across to you. Okay, this was done. Now, I'll tell you exactly what happens, okay. I'll tell you exactly what happens in your AIDS. So, students, let's say HIV is affecting the host, okay. What it is going to do? The first step for it to do is going to be from the membrane, the receptor is going to be there, okay. The receptor is going to be there. To the receptor, it is going to attach. Once it attaches, okay, let's draw the attachment. Students, with diagram only, this is the best way to understand, okay. I can chuma put one diagram and explain it to you, but it will not give you this kind of understanding, okay. Let's say to the CD4 receptor, your GP120 has attached, okay, GP120 has attached, 120 and CD4, okay, as attached. Now what is going to happen in the next step? Okay, in the next step, the entire capsid is going to enter inside. So this is what has happened. Attachment happened, right? Attachment happened, and immediately what has happened, students? The entire capsid membrane, okay, the entire capsid membrane has entered inside, okay, the entire capsid membrane has entered inside. Inside the capsid, what is there? Inside the capsid, what is there? Guys, I can see that most of you are not marking your attendance. Why so? Why so? Okay, is it so difficult to, appreciate the teacher? And I am drawing everything so that you people understand with me, okay. Okay, look here, students. In the second scenario, what happened? The entire capsid got inside to the host cell. Now, what is our next job? So, I am going to write the steps given in your NCERT, okay. So, the first step was to attach, okay, to bind to the receptor, to bind to the CD4 receptor. Clear? The second thing was the release of your entire capsid inside the host cell. Inside the host cell. This is the second step, okay. Second step has happened here. Now, what you have to remember, the moment this happens, that is where the capsid has come inside the host cell, what it is going to do? The first step it is going to do is convert, okay, convert its RNA to viral DNA, okay, viral DNA. Why it has to do this? Okay, it will enter the nucleus, and what it is going to do? Convert its RNA to viral DNA so that it can manipulate the cell. So, students, what exactly virus job is? It will enter inside your cell and it will tell you what to do. It will tell you what to do. Understood? So the next task is going to be convert its RNA to viral DNA with the help of whom? All of you tell me in the chat box. Whose help are we going to take? Okay? Okay. Jingle Lala, very nice. Okay. So tell me which enzyme is going to do this job? Ma'am, it is so obvious. It is going to be reverse transcriptase. So first we have utilized one. Reverse transcriptase. Okay. Reverse transcriptase, and what we have done, we have formed the viral DNA. Now let's assume this is the host DNA. Okay. Let's say this is the host DNA, and I'm doing a small partition like this, assuming this is the nucleus. Okay, nuclear membrane. This is the host DNA. Okay, I'm writing it here, host DNA. So in the next step, this step is done by the virus. Okay, this step is done by the virus, done and dusted by the virus. Now the next task is we are going to take the help of integrase, and we are going to add this. Okay, let's say, okay, to this, which color do we make, students? Pink only will keep it. Can you understand the difference? It's visible only, no? So into this, what happened? Integration happened. Integration of viral DNA with host DNA. Okay, every step I am teaching you so that when you see the diagram, you will be clear, okay, with the host DNA. Here, which enzyme are we going to take help of? We are going to take the help of your integrase, okay. Integrase is going to do this big job for us. Clear? Now what has happened, students? Viral DNA has got into the host DNA. Okay, host DNA. Now from there, the next step is going to be what? Form the viral proteins. Right, now DNA has happened. Now what will happen? The central dogma has to continue. Now we have to form our viral proteins and form more viruses, right? So viral protein will be formed. That time we will take the help of protease. Okay, from viral proteins, your new progenies, right? New viruses will be formed. Okay, everywhere more new viruses will be formed. Clear to all of you? Okay, new viruses will keep forming. Now see here, students, here one catch is there. The catch is that, okay, the catch is that if this whole thing is happening inside the macrophage, okay, macrophage is going to act as a factory, okay, for forming the new HIV. But let's say same scenario has happened with the help of T helper cells. The HIV exactly knows that T helper cells is a part of your cell mediated immunity. So what it will do, it will kill the T helper cells, right? It will kill the T helper cells, so your immunity is going to decrease. So in the acute phase, there will be presence of HIV. In the chronic phase, your immunity will drastically reduce. Your number of CD4 receptors also has to reduce. Now in the last, in your fatal stage, where you know you're able to understand that a small flu also you're not able to, like, your immune system is not able to cure. That is when we realize that HIV has multiplied in your body to such a level that now you are AIDS positive. Clear to all of you? You are AIDS positive. So this whole thing is clear. Now I'll show you NCERT diagram, but before that, just tell me if this whole thing is clear. If you have any doubts, ask me right now. Okay, ask me right now. I'm writing here macrophages, okay. I'm writing the cell as macrophages. Okay, so that we'll understand that, okay, CD4 receptor is there. Now let's say this capsid, okay, has entered into the host, okay, and this is your host DNA, nucleus is there, same thing has happened, this is your binding happening here, okay. Clear? No doubts? Now shall I show you NCERT? All of you tell me. Students, protease is also going to help in the breakdown of the protein code. I've seen it some places, okay, breakdown of the protein code, so we can manipulate the host, okay, host proteins as well and make our viral proteins. Clear? So breakdown of the protein code also will be done with the help of a protease. Now let's go to your NCERT and let's see the diagram. See, all of you, see here, I think it's visible, no? So here you can see that the retrovirus, viral RNA core, okay, viral RNA core, now viral protein coat, okay, virus infects the normal cell. Now from here, okay, one second. I think I have to click on this. Okay, now see here students, virus infects the normal cells. So as I showed you, now it has bound with the CD4 receptor. Immediately it is going to release, see, viral RNA is introduced into the cell. Clear? Viral RNA is introduced into the cell. Now once it enters, the viral DNA is produced by reverse transcriptase. So from RNA, single stranded RNA, we form the viral DNA. Now this viral DNA will be incorporated into the host genome, okay. So till now everything was happening in the cytoplasm. Now we are going to enter into the nucleus. Now this viral DNA will be incorporated into the host genome, and what is going to happen? It is going to manipulate your host, host nucleus, telling that, you know what, you should work according to me, okay, I am the king, you are the slave. So that kind of technique will happen, and that is why virus is so difficult to deal with. So here what is going to happen? See, new viral RNA is produced by the infected cell. So central dogma is continuously happening in our body, but without knowing that we are forming viral RNA, viral protein. We are not forming the host proteins, host RNAs. Clear to all of you? Yes or no? All confused people, students, so much effort and you people are not appreciating it, okay? Yes or no? Quickly, yes, very good. Now look here, new viral RNA is introduced by the infected cells and new viruses are produced. Clear to all of you? Yes or no? Very simple process. Just remember, the structure is very important for you to remember, and new viruses will start infecting the other cells. So from your acute stage we are moving towards your more chronic stage. From chronic stage it's going to get into a very fatal stage, okay? This is clear? Any doubts so far? Okay, if there is no doubt, let's proceed. Okay, let's proceed. Now the next question is, ma'am, exactly how does AIDS spread? Is it because of something else? Okay, so we will see that the mode of transmission, okay, mode of transmission, that is, how can a person be affected by AIDS? First thing first, students, it is by contact of, that means any fluids in our body, let's say blood. Okay, now blood, let's say you have a cut, okay, or one person has a cut, and I always give the example of this, boys go for shaving and trimming and everything. What happens during shaving? Let's say by mistake the salon guy has cut the face, okay, cheeks. Now blood has come. Now that blood hasn't come in contact with the shaving, okay, your Gillette blade. Now immediately the next person who is going to come, you will start, the person will start doing. Imagine this person also has a cut and by some means this has entered inside. By that also it can happen. That means through a cut, directly by in contact with the blood, the person can have a cut. But it's not necessary that it comes with the help of, a mosquito bite. Mosquito bite will not actually transmit HIV because of the main reason that mosquito, once they bite the person, after few minutes, okay, from the research I'm telling, once let's say mosquito bit a person, okay, sometime the mosquito is going to take some rest, and what they are going to do, they are going to digest the blood, because blood is their meal. So blood meal we tell, when I teach you malaria also you will come across the word blood meal. What is the meaning of that? That means the person, let the mosquito bit, after some time the mosquito is going to sit and digest that food, digest that blood, okay, and even if something is there on the, let's say on the at the tip of this, what is going to happen? That is going to get coagulated, okay, because blood will not stay, right, without coagulation for a longer time. So that blood which is on the tip of the mosquito sting also will get coagulated. So mosquito is not a reason for AIDS getting transferred, okay. Many, like, informations are there that mosquito bite will cause HIV, it will not. Clear? So you don't please scold mosquito for that. Now students, first thing could be a person could have AIDS because of sexual intercourse, that too unprotected sexual intercourse. Okay, unprotected sexual intercourse. Okay, unprotected sexual intercourse. Clear to all of you? Yes, mosquito will digest the blood. Yes, because it's a blood meal. Okay, blood meal. Okay, second thing, it can come through, as I told you, contaminated blood. Okay, contaminated blood. See, this is not infectious. Okay, this is not infectious, that AIDS, if you touch someone, it is not going to come like that, or through droplets. See, even in saliva, all of that, some amount of virus will be there, but it is so negligible that it cannot cause any spreading. Okay, so it doesn't, you do not have to get scared when you shake hands with a person who is AIDS, okay, who is AIDS positive, because it's not going to spread in that way. So contaminated blood, students, it can also be prenatal, that means from the mother to the child. Okay, from the mother to the child, it can come this way. Okay, and by blood transfusion, so when we are, you know, taking blood from blood banks, or when we are doing blood transfusion, it can happen if the person's blood is not tested. Okay, and from blood banks. Students, blood banks, it happens a lot because these people preserve blood from anyone and everyone, because they are marketing strategy. It is, you know, that okay, we have to be the blood bank that always have all the types of blood. So they will not test, if, and especially people do it in rural areas, that, you know, when they need, okay, money, some money for, let's say, alcohol, for other things, they actually donate their blood. Now what happens is, definitely a good blood bank will do all the tests possible, then only collect the blood and preserve it, right? Some people, what they do to do illegal business, they will collect the blood from an alcoholic person whose alcohol wouldn't have come down only. Okay, they will take blood from that person, or they take blood from person who are AIDS positive, any kind of diseases in the blood, they will take and they will transport these things to the urban areas or to, you know, big places. So it is always needed that we remember that when we are going to a blood bank, we always go to an authorized one, which the hospital itself might be connecting us directly to. Okay, what do you mean? Okay, yes, I did not understand the question, Tanuja. Okay, I think slowly you can type the message, by then I'll tell you. Students, by sharing the needles, okay, sharing needles. Now let's say, see, in this chapter only drug abuse concept is going to come. So you are sharing needles, okay, not you people, let's say generally I'm telling, there is drugs, okay, and all of you have seen intravenously they take in the drugs, right, they inject it to the veins. Now what happens, the needle is coming in contact with another person's blood. Because all of them are in a hallucination, all of them are sharing the needle during that time, there are chances that if one person is AIDS positive, the other person also can be AIDS positive, okay, because directly the blood has entered, the transfusion has happened, or the blood has mixed up, okay, that can also lead. So sharing of needles, this can also happen in hospitals, okay, it's not necessary it only happens during the drug thing, it can also happen in hospitals, it can happen in tattoo places. Okay, tattoo places, because why? In tattoo places, the person is coming directly in contact with the blood, right? Now even I have got a tattoo, but we have to make sure that the needle that they are using is a new needle. Everything they show that, okay ma'am, this is the needle, this is hygienic, they show. Okay, usually if you go to a very good place, they will show you everything that they are going to use, including the equipments, cotton, you know, the sanitizing thing, whatever, everything they will show right in front of you, okay? So that is very important for you to know, that when you are going to a tattoo artist, do not go to cheap places where they do it on the road, where they do it, you know, in some mela and exhibition they do, not the fake tattoo, I'm talking about the real tattoo one. Before times they used to do it with the help of I forgot the plant name, with one of the thorns of one plant, okay, but now it's stopped, it's no more done like that. So what they do is, it is coming in contact with your blood. So you should be careful. Clear? Right? Yes? Arohi, questions will come at the end of the session, dear. Questions will come at the end of the session, okay? No doubt in this, right? All of you are clear? Mechanism is clear? Yes or no in the chats? Okay, now this is all done. So how do we protect from this? Obviously we do not do any of this. Okay, whenever we are doing any of this, we always check and make sure that everything is properly fine and there is no mixing up of blood or anything like that. Okay, that is how we do it. Clear to all of you? Now students, the next thing is, ma'am, as you told, acute, chronic and fatal, what exactly happens? Remember students, in acute case also your lymphocytes, right, your lymphocytes will be decreasing. Now when I am talking about lymphocytes, think about your T cells, okay, T and B lymphocytes, remember. So your lymphocytes is going to reduce in number, and during the acute stage there are chances that, okay, high HIV content will be there, okay, high HIV will be there, content will be there. Why? Because in the initial stage they are going to be full, full-fledged, because the body is still trying to understand what just happened. Okay, I forgot about drinking water. Thank you for reminding me. Okay. Yes, thank you. All of you also drink water. So high HIV content will be there, students, during the acute stage. Then again in the chronic stage, same thing is going to happen. T helper cells are going to decrease in number. And do not think that, okay, as soon as we go, they are going to give you an antiviral drug. It's not going to happen like that, okay? Because first you have to do a preliminary test. So I'm going to talk to you about the test. So students, in the initial stage, you will do a preliminary test, which is your ELISA. Okay? ELISA. What is the full form of ELISA in the chats? If I tell you everything, how will it be? This is revision class. All of you tell me in the chat, what is the full form of ELISA? This is a preliminary test done to understand if the person has HIV virus. Quickly in the chats, let's see who's the first person to tell. I'm waiting for the first person to tell. All of you know the answer, yeah? It's not so difficult. We have discussed this before. Before in our life, we have discussed this, right? Yes, she is a good alarm. No? Tell quickly. Enzyme, something like enzyme. Okay, thank you, gamer. Something with enzyme. Enzyme linked immunosorbent assay, right? Enzyme linked immunosorbent assay is the full form of this. This you have to know, okay? This you have to know. Enzyme linked immunosorbent assay. I'm teaching very slowly so you understand, okay? Otherwise, I'll teach fast. Now, immunosorbent assay works on an antigen-antibody binding, but you luckily do not have to learn a lot about it, but just know that it is an antibody, okay? Antibody antigen binding process, okay? It works on this principle only. This is the principle of your ELISA. Now students, ELISA is a preliminary step if you want to go for further understanding, because there are chances that sometimes it can be fake positive. Okay, very good. Who told that? Newton, Sweetie, Tanuja, Madhumati, very nice. Okay, students, there are chances, okay, there are chances that during ELISA, when we did the ELISA test, there are chances that the results are negative. That means, let's say fake positive results can also come. Okay, fake negative results also can come. Now, this will cost you somewhere between 2,000 to 3,000. I don't know right now how much it is, but then that is the usual range of it. Okay. Now, there are chances that during this, let's say a fake positive has happened, you can go for a next test which is called as Western blotting. Okay, Western blotting, which is a little more expensive process, and this will give you most of the time, 99% time, the results are going to be true in this. Okay, Western blotting technique. This is also a test to understand if you are HIV positive or AIDS positive. This will give you most of the time 99.99% accuracy that okay, this is the reason. Okay, this is the thing. Now otherwise what happens? Now during your acute, during your chronic, you will start noticing something like, you will start noticing flu like symptoms. Okay, like you will become suddenly weak. Okay, you will have sudden weight loss. You will notice that you are sweating for no reason, or you're you have a small cold and it becomes very infectious. Like that, small things can happen for all of you. Okay, so Western blotting is the next technique. Now students, for the cure, let's say if someone is tested, ma'am, what would be the thing happening? For that we will be doing, we can give antiviral therapy. That is, antiviral drugs can be given. Okay, antiviral drugs can be given. And this could be like your many drugs are there, but you don't have the names. Okay, you do not have the names. Okay, yes. How can we know that it is fake? Yeah, that's what I told you, Eliza, sweetie. You can check after that. You can also check your you can do Western blotting. Clear to all of you? You can also do what? Western blotting. Okay, so students, there are different antiviral drugs. If you want one example, I'll tell you, that is AZT. Okay, AZT, shall I write it down only? Okay, AZT, which is called as acetylthymidine. Okay, acetylthymidine drugs. Again, this will not be given in the initial stages. Please do understand, they expect that your CD4 receptors number, okay, they check the CD4 receptors number, and once the number is up to this much, in every place it will be different. Now in India it will be different, now in South Africa it will be different, in South America it will be different. Depending on the CD4 receptor number, they are going to tell you that if you can take an antiviral drug or not. Chumma, if you go tell that, oh, I am HIV positive, they will not give you, like how you get Dolo in the medical store. No, you are not going to get that, because in government hospitals there are proper places where you will see that there is an HIV counter. There if you go and show your results, they will give you the antiviral drug. But again, it depends on checking everything properly. Is this clear to all of you? Yes, clear to all of you? So this is your identification, how you will understand diagnosis, and this is the prevention method. See, prevention, there is no prevention, but there is one thing, that you can increase the lifespan. Like let's say this person is going to die in 2 years, by the help of giving some drug, maybe little more extension will happen. Okay, there is no complete cure for this. Clear to all of you? Any doubts in this? Please let me know in the chats. I am waiting for all of your to tell. Okay, now students, prevention, many places like your NACO and your NGOs, many NGOs, they have all tried to get the awareness. Okay, try to give the awareness to people. So I will do one thing, I will just write here, because full forms can be asked. Okay, full forms can be asked. Okay. Okay, that's a nice one. Okay, soil and dust. Yes, so prevention in these kind of diseases, no, prevention is better than cure. So always be vigilant wherever you are, with whomever you are in life. Always be in a very alert situation. Do not fall for being cool, as I always tell, especially in youngsters, students. Drugs is the biggest problem. Other things you forget. Okay, drugs is the biggest problem, because everyone in the whole era of looking cool, people start doing drugs. Now what happens when you do drugs, right? If there is a small amount of blood also, that blood is going to have HIV, and it is not necessary that you have met that person in the acute stage. You might have met the person in a chronic stage or in a fatal stage. So maybe every drop of the blood will have enough HIV virus to infect you. So do not go for all this cool things. Please be normal. Normal is cool. Okay, so prevention, students, done by your NACO. They have been doing a lot of awareness programs, like in TVs, and as well as they are going to different rural areas and telling the people that please do not do unprotected sex, and they are also giving them condoms for a very small price. Like that, you also saw Saheli. So all that they are trying to spread. Got it? Clear to all of you? Yes, no doubt. So NACO's full form, you have to know. What is it? National If I go, so this much, okay, I cannot see the screen, but I hope you people can see. Okay, National AIDS Control Organization. Okay, control organization. What are they doing, students? They are bringing awareness to people, telling that you should not have, you know, sexual intercourse with multiple partners, especially when you are taking, you know, blood from blood banks or from very, you know, small hospitals. Make sure that you are checking the blood that you're taking and the blood groups and everything, okay, if there are any issues. So NACO is doing that. With that, many other NGOs also have tied up to bring awareness, okay, to bring awareness programs. And how are the awareness programs happening? Obviously with the help of, you know, visual, at the same time using posters, also giving, you know, talks about this. How is that awareness program happening? Okay, at the same time, you know, use of contraceptives also has been, you know, emphasized. Use contraceptives, okay, contraceptives have been emphasized by these programs. Okay, they use posters, okay, visual way of showing this. Clear? And which is the day that we celebrate the AIDS Day is going to be your December 1st. Okay, December 1st is AIDS Day. All this question can come sometimes. For your luck, if it comes, good, no? Yes? Clear? I think gamer, this speed is okay for other students. I cannot teach in more fast because I'm writing and teaching, no? So I think for you, you can watch it later in 2x. Okay, December 1st, AIDS Day. Students, speed is fine only, right? Clear to all of you? Done? Yes, it's an awareness day. Very good. December 1st, World's AIDS Day. Clear? Everything we have discussed. So students, you understood. And also let me talk about the incubation period. As I told you, in some people, see, there is no particular incubation period, but according to your NCERT, they have given 5 to 10 years, right? Incubation period is going to be in a span of 5 to 10 years. Now 5 to 10 years is not fixed in everyone, because as I told you, from the time that person is HIV positive, okay, it might take them a very long time to become AIDS positive. Okay, it might take a span of 5 years, it might take a span of 10 years, up to 20 years also it can take. Clear? So this span can be varied for different people. Clear to all of you? Done. Okay, now shall we go to cancer? Any doubts in AIDS? I hope all the questions are clear. So students, structure is done. Okay, structure of AIDS is done. Here entry, how they are going to enter and what is going to happen is done. Okay, here we have spoke about the mode of transmission, or you know, how it can be protected. See, all this if you take care only, protection also has happened. Then we have also spoke about how you can diagnose, how you can understand. Okay, clear to all of you? Then now we will talk about cancer. Clear? We took good amount of time to understand AIDS. Now let's learn cancer. Ready all of you? Full energy is there? No energy drain, no? Yes, drink water all of you in that time. Yes. Thank you so much. Okay, now tell me about cancer. Whatever you people know about cancer. Let me see what and all you know. Energy is high? Very nice, very nice. Okay. Now students, cancer, simply from one word itself we understand, no? Normal cells. Let's first draw some normal cells, okay? Today I have decided that I am going to teach you with more diagrams, okay? More diagrams, because most of my students have really good visual understanding. If I draw the diagrams, no, some of my students tell me, ma'am, I still remember that, you know, when I was writing NEET exam, I remember that you drew this in pink color. So that is why I am drawing the diagrams, okay? Yes. So students, let's say this is your normal cell, and let's also draw your cancerous cell. Okay, let's draw your cancerous cells. Yes, this is your cancer cell. So these are the two different cells, okay, clear to all of you. Now, what is the biggest difference? So students, this one is your normal cell, and normal cell has one major thing that is called as contact inhibition. That is, when one cell comes in contact with the next cell, it knows that, okay, I have to stop my division, it's over. Okay, I do not have to do anything else. But cancer cells, what is the problem? cancer cells, the problem is that they have lost the ability of contact inhibition. That means normal cells show contact inhibition. This is very important, okay? This word is very important, contact inhibition, okay? And this cancer cells have lacked, okay, the ability for contact inhibition, okay, for contact inhibition. Now you tell me, contact inhibition is stopped. So don't you think they are going to do continuous cell division? Yes ma'am, they will do continuous cell division. Why? Because I am standing here and another cell is here. The moment I touch the cell, I know, okay, I have to stop division. Cancer cells are not like that. They will hit this cell, they will hit this cell, they will move everywhere. They will spread as much as possible. So normal cells, contact inhibition is there. Your cancer cells, contact inhibition is not there. Clear to all of you? Okay, what spelling and all you people type, yeah? I feel like crying sometimes, okay? Yes. Now, this cell, normal cell, have a proper definite lifespan, okay? Definite lifespan. That, okay, you will live till here and you will die till here. Proper definite. This is not like that. No definite. Students, it's important for you to know the difference between normal cells and cancerous cells, okay? No definite lifespan. No definite lifespan. Then here what? Here it is normal, okay, proper regulated division is going to happen, okay, everything is going to be regulated. So, regulated cell divisions, okay, divisions, and here what is going to happen? Uncontrolled division is going to happen, okay, uncontrolled division is going to happen. All these notes will be available on Vedantu NEET English Telegram and Biology by Gopika. Clear? Uncontrolled cell division. Can all of you tell me any other thing that you remember? Anything else that you know from normal cells and your cancer cells? It could be anything that you know. Come on, all of you tell me anything, something that you know, do not know. Okay, how much is the target? Have we reached? No, we have not reached. Okay. And most of you have asked for the timetable. I will be releasing the timetable, okay? I saw a lot of comments on the timetable, so definitely timetable is going to come soon. Okay students, this is clear. This is your cancer cell, this is your normal cell. So basically, these people can become a mass of cells. A mass of cells we also called as neoplast, okay? Neoplastic, we tell this words we will see later. So students, remember that a mass of cells, we can also call it something called as tumor. Tumor. That means, let's say, this is a lot of cells, okay? From one cell, many cells proliferated and formed a mass of cell, and this mass of cell will be called a tumor. Okay, so if you see the word tumor, do not get confused. So you ma'am did not talk about this only in cancer. Tumor is a mass of cells, okay? Mass of cells. Clear? Done. Cancer, general idea understood. Difference between normal cells and cancer cells, if you know, half of your problem itself is solved. Okay, half of your problem is solved because of this. Okay. Sorry. Yes. Very nice, very nice. All of you are understanding. Happy for that. Okay, happy for that. Okay. Tomorrow you want the timetable. Huh? Okay, tomorrow means tomorrow, today means today. Okay, I'll give you. Okay, so this tumor, I told you, neoplastic, okay, or we can also call this as neoplasm. So I'll just write it here so that when I'm writing later, you'll not have the confusion. You, from where is this ma'am tell? Okay, neoplasm. Done. Now students, two types of cancer is there, okay, depending on how it is, two types of cancer is there. What is it called? Benign cancer and your malignant cancer. Cancer is of two types, okay, benign cancer, sorry, tumor, benign tumor and malignant tumor. Okay, malignant tumor. Now tumor you understood, no? A mass of cell is called as a tumor, okay? We can also call it what as neoplasm. Now students, two types of tumors are there. One is called as benign tumor, one is called as malignant tumor. Let's draw some set of cells, okay? Let's draw one kutty box. In that box, let's just draw few cells for you to understand. Looking at this, you tell me what do you understand, okay? What do you understand? Clear? Now let's say students, I am drawing some cells inside this. Okay. Just tell me, looking at the diagram, what do you understand from this? Don't tell me you don't understand anything. That will be such a lie, because it's my students I'm telling. Okay, this is your one set of cells. Okay, this is one set of cells. I wish I had the option. Okay, ma'am, doubt, ma'am, chapter weightage. Don't know, dear. Okay, how many times I've told the chapter weightage? I have forgotten that number of times I've told. I've also done a dedicated video on chapter weightage. Still, you people keep asking me, chapter weightage, chapter weightage. Okay, so I cannot draw the whole set of cells. It will consume a lot of time. So students, just assume, okay, so many cells are there. Okay, just look at the diagram and you have to tell me what do you understand. I think all of you know, but those who are studying this for the first time, it will be very helpful for you to understand. Okay, let's say I'm not drawing the whole thing, but just tell me what do you understand. Okay, what do you understand from this diagram, students? Tell me. Understanding is understanding. Okay. Okay, yes. Okay, tell me this. From the diagram, what do you people understand? My students are very funny. Okay, from the diagram, ma'am, I understand that you have drawn a beautiful diagram, but no, not that. Look here, students, your benign tumor is actually, it is completely in one place, right? Yes or no? If you look at this, don't you feel like it is remaining confined to just one place? So the first most important thing about your benign tumor is that it is confined to one single place. Okay, it is confined to one location. Okay, okay, confined to one location in your body. Let's say if a person has, okay, only on the breast, okay, or only on the arms, okay, somewhere in the arms, or let's say somewhere in the body, it will be confined to that one place. It is not going to move any place. So this is actually less dangerous than your malignant tumor. Understood? Yes, it will be confined to just one location and it is going to be less dangerous. I'm not telling it is completely, not dangerous. It can be painful for some people. So it is less dangerous compared to your malignant tumor. Okay, comparison to your malignant tumor, I am writing empty. Okay, malignant tumor. And the second thing, students, third thing, it can be painful, but most of the time, majority of the times, it is not painful. Okay, it is not painful. So I am writing here majority of the time. Okay, major times it's not painful. Clear? Yes or no? Easy peasy lemon squeezy? Yes. Not spread to other places. Okay. Now, when I talk about malignant tumor, as you know, it is very dangerous, right? The term itself is dangerous. So, malignant tumor is not, okay, confined. It's not confined and it will spread to other places. Okay. It will spread to other parts and form your secondary tumors. Okay, other parts and form your secondary tumors. Done? Now, there is one word called as metastasis. Okay. Okay, so look here. Oh, that's so sad. Money. Okay, now students, spread to other parts and form secondary tumors. Now, the condition, okay, we can use the word called as metastasis. What can we call it as? metastasis. That is, from one place it proliferates, okay, and goes to another place and it starts spreading there, okay. So metastasis is where, like students, all of you remember hair, your hair, no, whenever, let's say when you're washing your hair, it will go from where you're washing, it will go to the sink where the water is flowing, it will go and get logged there. Okay, now it's staying there only. It's like benign tumor. It has gone there. It has stayed there. But let's say the hair is in a, you know, in a big water body. What it will do? It will go wherever it goes. It will go and make the block. Okay, wherever it will go and make the block. So here they are going to do metastasis with this. Okay. Oh my God! What and all you want to know, gamer? I think you should just play games, right? Yes, tell me. So many doubts. Yes, okay. Now, there is also a term called apoptosis. Apoptosis is programmed cell death. That's why I told you when I spoke about the lifespan only, right? Here I told you they have a definite lifespan. Here it is not definite. So, here only I can write apoptosis is absent. Okay, apoptosis is basically programmed cell death. The body will decide that, okay, don't, this much only, this is just enough. Okay, so apoptosis, programmed cell death, I am writing it here only so that all the terminologies are clear to you. Okay, programmed cell death. Okay, now students, yeah, this metastasis is clear now to all of you. Yes, any doubts in metastasis? If you have, please ask me right away, okay? Yes, clear? No doubts, right? Yes. Thank you. Thank you. Okay, students, this is done. Okay, fine. Now, there is one term called as angiogenesis, where it is very simple, students. Let's say this is your, you know, cancer causing cells. Okay, this is your cancer causing cells. Now, what they will do, no, they're so like chalak, okay, chalak people they are. What they will do, students, they will start forming blood vessels around them. Okay. New blood vessels will start forming around these cells, because of which what will happen, other cells will be not getting sufficient nutrients. So, other cells will go through a, you know, they will start dying. But the cancer cells, to be so smart, they are like, I know what I want to do. So, what they will do a angiogenesis, that is where they will start forming the new blood vessels. Okay, formation of new blood vessels. Okay, very chalak they are, very chalak they are. They will do angiogenesis. So, what is going to happen here? You will notice that near the cancer cells, there is a growth of new blood vessels. Okay, new blood vessels will start growing, and because new blood vessels start growing, other cells will become nutrient deficient. Okay, nutrient deficient. So, new blood vessel formation, okay, new blood vessel formation is going to happen, leading to the other cells to go nutrient deficit. Now, malignant and benign tumor is clear to all of you. Benign tumor is confined to one place, malignant tumor is not confined to one place. Okay, they are going to spread. Now, malignant tumor is more dangerous one, because this people can become very fatal. Okay, they are very dangerous, very dangerous. Okay, and they can be painful most of the time. Not most of the time, usually they are very painful. Okay, very painful. All the points are clear. Don't ask me later doubts, now only tell. Okay, yes, fine. Now, this is your cancer, benign tumor, malignant tumor. Here the properties I have written. Okay, students, these are actually properties. Okay, properties of your cancer cells. If they ask you properties, don't be like, oh, you ma'am did not tell the word properties. These are the properties. That means what are they different? How are they different from the normal cells? That is the properties. Be it apoptosis, be it metastasis, okay, and angiogenesis, indefinite cell division, all this. Programmed cell death is absent. Last time I actually taught you the types of cancer, but I do not see any questions based on that. So, I am just doubtful that if I should, actually teach you that. Okay, it is nothing but, carcinoma, sarcoma and leukemia, but like just as terminologies you can just know. Shall I just write in the corner? Okay, I will not go into the depth of it like last time I went. I will just write the 3 types, okay, based on how the cancer is going to happen. 3 types are there, okay. So 3 types. I'm just writing the word, okay, so that when you see it somewhere, you shouldn't be like, oh, you first time I'm seeing. Carcinoma, sarcoma and leukemia. Okay, leukemia. So these are the 3 types of cancers, okay. Now again, germ layers, all that will come under this, but just know this for the time being, okay. Got it? Yes. Where have we learned the word sarcoma? All of you tell me. Something to do with sarcoma. Who remembers anything? Okay, students, in the muscles we have remembered rest, yes or no? Now MK Ansari is asking, ma'am, what is this? Sarcome, okay, sarcomere. So something to do with your muscular tissues, okay. It can cause a problem in your muscular tissues. That is a cancer caused by in your muscular tissues. So is this clear? Now shall we talk about how the prevention is happening, how we can, like, how we can protect. Now students, remember that cancer identification and prevention and causative all is separate in your textbook, right? Separate in your textbook. If I go back to a textbook, you will notice that in your cancer, I will just show you how your textbook it is shown. So one second. See, prevention of AIDS they have given. See, cancer, if you see below, so students, here tumors are given, contact inhibition they have highlighted. Now here you see causes of cancer. So first they are starting with causes of cancer, where they are talking about your physical, chemical and biological agents, which I will teach you right now. Okay, as you go down you will understand detection and diagnosis, and most of the students what happen, they get confused with this, and then when you go down, treatment of cancer. So most of the students get confused with treatment and diagnosis. First is identification, how to know that this person have cancer, okay. Now this is going to be treatment, how we can treat cancer, okay. As all of you know, your chemotherapy is there, radiotherapy is there, immunotherapy is there, so all that only. But here you have MRI, CT scan, X-ray scan, biopsy, histopathology, all that here we are going to discuss, okay. Fine, clear to all of you? I have shown you NCERT as well. Now let's first start with agents that are going to cause cancer, okay, like how cancer is going to be caused. Clear to all of you, yes or no? Cancer, how it is going to be caused. So look here, let's say this color is not even visible, students. Yes, here, okay. Now students, it can be caused according to your NCERT. They have given 4 ways to how cancer can be caused. First is going to be your physical method, okay. First is physical, okay. Next is chemical, three ways, okay, sorry, and third is going to be biological. Okay, biological. These are the 3 ways, okay. Clear to all of you? Yes, very good. I think someone told muscles, very nice. Philo told about muscles, very good, okay. Yes, Philo told about muscles, okay. Now students, all of you like the video quickly. Okay, now students, physical, okay, physical. Now what do I know? See that, let's say too much exposure to X-rays, okay, any ionization, right, X-rays, gamma rays, that can cause cancer in our body, okay. Too much exposure to what? X-rays, gamma rays, all of this can cause us cancer. All of this can cause us what? Cancer. That means any ionizing, any radiations for that matter can cause cancer, okay. I'm just writing here radiations, these are also radiations only. I'm just writing here radiations, okay. Okay, ionizing, got it? Yes, clear? So, physical, X-rays, gamma rays, any ionization, any radiations can cause, but how does it happen? I will tell you when I am talking about biological, okay? Right now, don't break your head for that, but right, yes, physical, chemical, biological. Chemical, students, how? Tobacco, okay, inhaling of smoke. In that smoke, if some, carcinogens are there, that can also cause. Nowadays, I can also say that some of the, products that we use on our faces, like let's say face wash, toners, sunscreens, those also have carcinogens in them, which are not highlighted by any companies, okay? But it is your job to check if they are having any carcinogens, okay? So always remember that when you are using something from, any local store, if you have bought something, check if there are any agents that can cause skin cancer on a longer run, because we use face wash for a longer time. Right, so it is important for us to remember that. Clear? Okay, so tobacco, smoke, all of this. Now, biological agents, very important. In our body, there are some inactive genes which are called as proto-oncogenes. These proto-oncogenes, students, can get activated, okay? Can get activated with the help of any of this physical or any of this chemical agents. Clear to all of you? So, biological agents, we tell proto-oncogenes which can cause cancer. Okay, they are present in our body. They will get activated. Now they are in the inactive form. Let's say because of the extreme exposure to X-rays, gamma rays, or any exposure to smoke, tobacco, carcinogenic, substances, this proto-oncogenes can get activated. Is this clear to all of you? Okay. Why in your keyboard C is not there? Huh? What is the cause of cancer in children's? I cannot understand your spelling. I cannot understand any of your spellings. Okay. Yes, is this clear or not clear? Tell fast. Okay, so what happens? This proto-oncogenes will get activated to become oncogenic. Okay, oncogenic, and then they will become cancer causing. Clear to all of you? Yes or no? They'll from inactive form, they will become active form. So, how did they become active form? It is because of the exposure to any physical or chemical agents, okay? Continuous exposure. Clear, sir? Clear, ma'am? Gender also my students have forgotten by the time they reach cancer. What if I would have taken the whole chapter at one shot? Okay, you would have forgotten your own identity only. Why am I here? Am I actually a NEET aspirant? What am I doing in life? Okay, so here, this is clear. Biological agent, very simple. 2-3 words is given in your NCERT. Inactive forms are called proto-oncogenes. They will get activated due to the presence of any race. Okay, and they will become oncogenic and they will become viral oncogenes. Clear? Clear? Yes. This is your physical, chemical and your biological. Okay. Yes, drink water all of you. Clear? C is visible in cancer, it seems. So, you people, how much you talk? Yes, okay. Here you can also write UV rays, okay? UV rays also, because they are non-ionizing. UV rays. UV rays. Yes. Students, UV rays is the strategy for sunscreen to be sold, okay? I am actually breaking many people's business here, right? Because they keep telling that you are exposed to UV rays, you can get cancer. That is because of this, okay? Done. Cancer, this is done. Now, next one. No place I left. Okay, 1 second, students. Prevention and cure is there. That also we'll finish it off, okay? After that, we'll take a lunch break. And something. sorry students, okay, done. Next is, how do we identify? Okay, how do we identify that some person has cancer? Okay, okay, it's okay. So here, who is telling you? See, people who don't have C in their keyboard is telling me about UV rays. Okay, listen. Now, how do I identify that some person has, you know, cancer? So students, we can do biopsy. Okay, many techniques are there actually. It's just for your knowledge. Okay, just know this. Biopsy we can do X-ray. Okay, we can do you know, histopathology, histological test. That means where we take a small, you know, portion of the tissue. Okay, this is our tissue. We check in that tissue if there is any changes. Okay, let's say the cells, you can understand, no, when there is a mass of cells, we will definitely understand. So we will check the histopathology of it. Okay, oncology is the study of cancer. Okay, so that's why oncogenes, no, don't get confused. You, where is this word oncogenes coming from? This one. Okay, clear to all of you? Yes, we okay, now tell me about this. Histopathology is clear, biopsy is clear, X-ray is clear, CT scan also we can do. Okay, we can do bone marrow test. So many tests is there. Okay, so many tests we can do to identify if the person has cancer. Yes, very good. I think someone told, Sony has told. Yes ma'am, blood and bone marrow. Okay, bone marrow is also a very common test. Bone marrow, I think Sony has my previous notes. No, Sony. Okay, bone marrow test. Okay, blood test, bone marrow test, then histopathological test, X-rays, biopsies, CT scans, MRIs, okay, PET, PET. PET is not given for you. So here, okay, CT, no, MRI scan. Students, MRI is more preferable. It is expensive, but the thing is, this is magnet resonancing imaging. So it is more, let's say, like, see here and all, again, even though we told that the physical agent to cause cancer is X-rays, and then we are only telling that do an X-ray where there is rays, right? So that's why what we have to do is, it is better to do MRI scan, even though it is on an expensive side. This is a, you know, magnetic resonance. Okay, this is magnet based, right? So magnetic resonancing image. Okay, resonancing imaging. So this is a more preferable one because you are not exposed to unnecessary ionizing or non-ionizing radiation. So that's why MRI is more accurate. Okay, like more preferable, MRI is more preferable. Yes, yes. Clear to all of you? Any doubts so far? Students, many scans are there. You can write, okay, CT scan, you write, X-ray, you write, 3D imaging, you write. But most important is when we talk about treatment, it is very important, okay? Treatment is very important. These scans are just given in your NCERT. Now, when I talk about treatment, how do we treat a person who is, you know, cancer, like we have diagnosed that person with cancer, how do we treat that person? See, you can treat that person with radiotherapy. Okay, radiotherapy. Luckily, you do not have to go into the depth of it. So, you just have to know the names. Okay, radiotherapy. Then what can we do? We can do chemotherapy, which is very common nowadays, and you people would have heard about it also, where the person lose their hair and everything, and it has become very common. Okay, where celebrities also are talking about it. They're showing their chemotherapy, you know, images. Not everyone, I'm telling, few people who have got, you know, cancer. So, chemotherapy, you know, where chemical radiations are passed to kill the cancer cells. Now you have radiotherapy, at the same time you have immunotherapy, where we will use our immunotherapy, where we will use your alpha interferons. Okay, interferons, because we know that interferons are released by your cancer, like a cell which has been affected by a virus. So they will release interferons, which are antiviral proteins, which will let the other cells know that, you know what, I am affected by a virus, you please, you know, take care of yourself. So alpha interferon techniques can also be used. Okay, immunotherapies can be used, radiotherapies can be used, chemotherapies can be used, and in cases of, let's say, a tumor where it is confined to one place, we can also do surgeries. Okay, we can also do surgeries. As you know, it's very common to do the breast cancer, where they remove the breast, right? Where they like remove the breast, in the sense you understood, no? Where the cancerous cells are there, to remove that. So surgeries also are possible. So these are the 4 techniques mentioned in your NCERT by which we can actually treat cancer. But as of now, there is no complete cure of cancer. Like we cannot tell that, okay, you take this tablet and you will be completely cured of cancer. That is not still existing. Okay, that is not existing, and I hope and pray that one of my students, one of the, any of the students are able to be in a position where they find drugs for cancer. See, I have read a lot about the research papers where they're telling that this drug is finally out to cure cancer, but there is no I wouldn't say there is no breakthrough has definitely happened, but there is no like one solution that, okay, this will definitely cure cancer. That kind of thing has not happened. Okay, clear to all of you? Yes? Clear? Some fire emojis in the chat box if it's clear, and quickly mark your attendance as well, so I understand it is properly clear to all of you. Okay? Yes? Yes, some anti-cancer drugs also have been mentioned to you people, but it is not there in NCERT is what I believe. Okay, which will basically, students, they will reduce the proliferation of the cell division. Cell division will be a little inhibited, okay, and they are extracted from your Vinca rosea, which I have told you in last class also. Okay, so they will not like completely cure cancer, they will just stop the cell multiplication. That's it. Okay, clear to all of you? Very nice. I think Matre, Philo, Madhumati, all of them understood. Sweetie understood. Yes, very good. Okay, that is your drugs. Okay, vinblastin and all of that. Those are one way of reducing or inhibiting the cell multiplication. Clear? Please don't write that, yeah, there is one tablet which will cure cancer completely. It will be such a blunder from your side. Okay, blunder from your side. Okay, inhibit. Okay, got it? Yes. Fine to all of you? Very nice. I can see a lot of fire emojis. Very nice, very nice. Perfect. Okay, perfect. Yes, clear? Okay. Now students, malaria I have to do. I think we will come back after a break. Is that okay? Right? Because none of you have had food also. Okay, I want all of you to have food, and at the same time, prevention done, treatment done. Okay, it's done. Okay. Now, okay, we will just go through NCERT also if we miss something. Oh my god, what is this? Yeah. Okay, proto-oncogenes. See, oncogenic viruses have genes called viral oncogenes. Furthermore, several genes called cellular oncogenes or proto-oncogenes have been identified in normal cells, which when activated under certain condition could be oncogenic transformation. Okay. Yes. Cancer detection and diagnosis, as you can see here, they have given that cancer detection is based on biopsy and histopathology studies of the tissue and the blood, and bone marrow test for increased cell counts in the case of leukemias. In biopsy, a piece of suspected tissue is cut into thin section, stained and examined under the microscope, and we will be able to understand. Okay, study of tissues, all of you know. So they will do histopathological studies by a pathologist. Pathologist will check it. Okay. Now, techniques like radiography, CT scan, MRI scan are very useful to detect cancers of the internal organs. Computed tomography uses X-rays to generate three-dimensional images of the internals. That means 3D images will be done, okay, using X-rays to generate three-dimensional images of the internals of an object. MRI uses strong magnetic fields and non-ionizing radiations to accurately detect pathological and physiological changes in the living tissues. Any doubt in this? Okay, now here important part, antibodies against cancer specific antigens are also used for detection of certain cancer. That means we can also do ELISA where antigen antibody is done, but here it is different. Here what is going to happen, antibodies against cancer specific antigens will be done. So it can be vice versa. Clear to all of you? Yes? Okay, now here that can also be done to identify cancer. Identification of such genes which predispose an individual to certain cancer may be very helpful in prevention of such cancers. Such individuals may be advised to avoid exposure to particular carcinogens. That's what I told you. How does your oncogenes become viral oncogenes? It is only because of the exposure to carcinogens. Okay, exposure to carcinogens. And students, here treatment, as I told you, same thing only. See, you can see surgery, radiation therapy, immunotherapy, as well as chemotherapy. Radiotherapy, tumor cells are irradiated lethally, taking proper care of the normal tissue surrounding the tumor mass. Several chemotherapeutic drugs are used to kill the cancer cells, but sometimes what happens that your normal cells also will die during these situations. Okay, and that can lead to hair loss and anemia. Most cancers are treated by a combination of surgery, radiotherapy and chemotherapy, and all of you know that there are stages of cancer, right? Stage 1, stage 2, stage 3 and stage 4, or stage 0, then 1, 2, 3, 4. Okay, so in stage 0 we will be able to do it with the help of surgery. We will be able to remove the tumor or the mass of cells immediately, and maybe I wouldn't say 100% everyone who has done cancer at the 0 stage have recovered from cancer, because the problem is the cells are so complicated that they could have spread. Okay, so that can happen. Now stage 1 little more severe, stage 2, stage 3 and stage 4. By stage 4 it is already very fatal. That means it has already taken over all your immune systems as well as all of your organs. Okay, so it is very dangerous during the stage 4. Now here you can see the patients are given substances called biological response modifiers such as alpha interferons, which activates their immune system and help in destroying the tumor, because they are antiviral, right? So they will know that I am being interferons are why I told you in cytokine barriers itself, that they will alert the other cells. Okay, yes. So here, yes, research has been happening on the stem cells as well. Okay, very good, sweetie. Stem cells also, they have been trying to find out if stem cells that we extract at a very embryonic stage or later in the stages, will it help in curing cancers. But as I told you, there is breakthrough, but there are there are small breakthroughs. There is no like proper breakthrough telling that okay, this is a scientific way of dealing with it. That is not there. Okay, students, is this clear? After this we have to go to common diseases. So I think another, yes, now what's the time? I can't even see the time. Yeah, okay. So another maybe and half an hour, we will be half an hour, 45 minutes, we will be done with malaria, and we will just go through other common diseases. Okay, is it okay for all of you? Will you come back after a lunch break? Can I expect all of you after a lunch break, or will you all run away? Okay, will you all run away? No one is going to run away, right? So we will come back. Students, I need time to have my lunch. Okay, so please give me some 20 minutes time, because I have just ordered, so it will take some time to come back. Sorry, food to come. Okay, resume back after. Okay, works. Yes, no running away, coming back. Okay, no running away, all of you have to come back by 2:20. All such they only my students are like this. Okay, they only tell the joke, they only laugh. Self-proclaimed comedians there. Okay, stand-up comedy, all the kinds of comedy they'll do, but no one else will laugh except them. Okay, yes, so see you all at 2:20. students, I'm back. Yes. Can we continue? Yes. Okay, let's take, yes, welcome all of you also back. Okay, did you all have lunch? Okay, my lunch took time, even though I ordered on time, it took some more time. So that's why. Yes. So students, AIDS and cancer, pakka clear, right? No doubts in AIDS and cancer. We are going to start with malaria, okay? Because as I told you, I'm only going to do the major concepts and from where questions have usually come, okay? After this, you can just go through your human health and diseases, like you can just go through the like, you know, other topics like be typhoid and all. We will just discuss through it, okay? Now students, malaria, it is very important. So sorry. Malaria, it is very important for you to know the life cycle. Clear? Yes. I think some more have to join. That's why I'm waiting for like 2 minutes so that others also join. And then we will start. Okay. Okay. Now students, malaria. Malaria, first of all, you have to know the causative agent. Who is the causative agent of malaria? Plasmodium. Plasmodium vivax. Yes or no? Plasmodium vivax is the causative agent of your malaria. Okay. Causative agent of your malaria is Plasmodium vivax. But there are other varieties also, like your Plasmodium ovale, falciparum, malaria. In that, falciparum is the most, okay, falciparum is the most malignant one, okay. Fatal one is going to be Plasmodium falciparum. Okay, this is the most dangerous one, fatal one. Got it? Question has come on this, that's why I am mentioning it here. Now, remember that when we talk about malaria, we have to remember few things, like what? All of you tell me in the chat box, ma'am, what do I have to remember about malaria? First of all, students, they are digenetic, okay? Digenetic parasite. That means they want two hosts, okay? They are digenetic, that means they want what? Two host. Very, very important for you to remember that they need your primary host as man, okay, primary host as man and secondary host as mosquito. Secondary host as mosquito. Clear? Now, which mosquito exactly? Female Anopheles mosquito. Okay, which mosquito exactly? Female Anopheles mosquito. Done? Clear to all of you? Yes, all of you? Yes, very good, very good. Okay. So, female Anopheles mosquito. So, digenetic, that means they need a primary host as well as a secondary host. Primary host, this question has come. Which is the primary host? Is it the man or is it Anopheles mosquito? Primary host is going to be the man and secondary host is going to be Anopheles mosquito. Okay. Ma'am, why always female? because male are just going to consume the meal, but these are the ones who are going to act like vectors. So students, I will call this a vector. Why am I calling it a vector, not a carrier? Because vector will have one of their life cycles. Okay, one of the life cycle is going to be inside the mosquito. That is why we call them a vector, and they are carrier of disease. That means they are going to carry the infectious disease, and also one life cycle is going to happen inside them. Okay, one life cycle is going to happen inside them. Now, understood that plasmodium ovale? Yes, ma'am. Clear to all of you? Yes or no? Okay, I can see that lot of fire emojis. Okay, now listen students carefully. Now you have to understand that in this life cycle, your human and mosquito, there are some steps that you have to always remember. Yeah, she is sheing, that's why. Now students, some steps are going to be that, or some words that we are going to discuss are going to be sporozoites. All of you tell me, what is sporozoites? Okay, I don't think I will be able to fit in the life cycle here, so I will just write the rest of it here. Okay. Anopheles mosquito. Okay, what are they going to have? They are going to, let's say they are going to bite an infected person. Okay, or they already have the plasmodium vivax inside them. So what is going to happen? They are going to infect, okay, bite a male. So I think students, I'll write this in the next page itself. Okay, we will write 2 tabular columns and learn this. Okay. Now let's say this is the life cycle that is going to happen in man. Okay, man. And this is the life cycle that is going to happen in the mosquito. Okay, mosquito. Same size, huh? I think it's too big. Yes. Okay, fine. Same size? Similar size. Okay. Now, look here students, we are going to write the life cycle. Okay, we are going to write the life cycle. That is where you are going to understand that what is exactly happening. How is it exactly happening? So, let's start with man. Okay, this is the man, that is the mosquito's cycle. Now, after that, we will see NCERT. So, what is going to happen? Mosquito is going to be there. That mosquito is already infected. Right, that mosquito is already infected. Now that infected mosquito is going to bite the man and it is going to release sporozoites. What are these going to release? Sporozoites into the body of the male. Now that sporozoites is going to go to your hepatocytes. Tell me, where is hepatocyte cells presence? It is in the liver. It is going to your hepatocytes, and there what it is going to do, it is going to burst your hepatocytes. Okay, so let's draw your hepatocyte cells, and students remember, these cells are going to burst open. Okay, starting they are just going to, like, you know, come there and be like, oh hi, but then later they will start multiplying, and they are going to do burst of the cells. Clear to all of you? Okay, they are going to release burst. This is your first. Very good. Liver cells. Very, very, very good. Okay, my students are so smart. Yes. So, hepatocytes it will go. In the hepatocytes, it is going to cause the bursting of the cells. Okay, bursting of your liver cells. That is because of continuous multiplication. Okay, continuous multiplication. Because the liver cells burst open happen, now where is it going to go? It is automatically going into the blood vessels, right? Okay, blood vessels. So it is going to get into your blood, and in the bloodstream it is going to go and attack your RBCs. Okay, clear to all of you so far is good? Yes, all of you are able to understand? Very good. Okay, now it is going to get into your bloodstream and it is going to attack your RBCs. So let's say from here it entered into your bloodstream. Okay, it entered into the bloodstream. Now it entered into the bloodstream, so it's understood that it is going to come and attack your RBCs. So let's draw your RBCs. In the RBCs also, what is going to happen, students? Obviously, it is going to rupture so much that the RBC, okay, is going to burst out. Okay, that question, Sony, no one has ever found an answer to that. Okay, okay, this question keeps continuing, no, like that. So here what is going to happen? Again, bursting, opening, okay, bursting of your RBCs cells. Okay, RBC cells. So this is your RBC cells. Okay, here also same thing is going to happen, where they are going to burst open. Students, when it is going to burst open in your RBCs, there is one thing that you have to remember very carefully, that when this happens, there is going to be a release of one person which is called as your hemozoin. What is going to be released? Release of hemozoin. Release of what? Release of hemozoin, which is going to show the signs of fever and chills. Okay, hemozoin is going to be released, which is going to show the signs of fever and chills. Okay, so here you will start, the person will start realizing that they have fever, chills. Okay, everything happens when the RBCs burst open, and that leads to the release of hemozoin. Very good, Madhumati. Okay, so that is the cycle that is going to happen in man. Now, ma'am, exactly what is happening? Students, basically in man, in the life cycle, we are helping them to produce the gametes. Okay, we are helping the plasmodium to form the gametes. Okay, so here in this whole process, gamete formation is what is happening, but fertilization, that means fusion of the male and female gamete, is going to happen in the mosquito. Clear? So here gamete formation has happened. Okay, I am writing it down. Gametophytes. Okay, gamete formation has happened. Now that gametes, be it male, both your male and female gametes, fusion will not happen in our the man. It has to happen in the mosquito. So mosquito is going to collect it back. Okay, it is going to, the next mosquito will come, it will collect the gametes back. Okay, it will collect the gametes back, and also understand hemozoin also helps in the development of the gametes. Okay, here first thing is going to happen is fertilization. Okay, fertilization of your gametes is going to happen. Clear to all of you? Now fertilization will lead to what? All of you in the chats, quickly. Fertilization will lead to what? That means here, let's say before fertilization, I will just write gametes will be there. That gametes are going to undergo fertilization. Fertilization will result in the formation of a zygote, and the zygote is none other than your sporozoids, right? The zygotes will mature to form sporozoids, and this sporozoids will be stored, okay, will be stored in the gut of the mosquito. So this sporozoids will be stored in the gut of the mosquito. Okay, in the gut of the mosquito it will be stored. Storage is here, students, but remember that when it is going to bite, okay, when it is going to bite, it is going to keep it in the salivary gland. That location you have to remember. Yes, all of you clear? Very good, okay. So it is storage is going to happen in the salivary gland, and the formation of this gametes will happen in the gut. Okay, so it is going to store it in the salivary gland, and in the salivary, after that it is going to bite the next person. So next it is going to again infect another person. Okay, clear or not? Yes, clear or not? Is it clear? This is clear. So what is going to happen? The mosquito got infected with Plasmodium vivax. Okay, so here the life cycle is of man. This is what is happening in man's body. This is what is happening in mosquito's body. Okay, that only they have given in a in mosquito's body. So here the mosquito got infected, that led to the release of sporozoids inside the body. That sporozoids went into the liver cells, and there it burst open the liver cells, and it got released into the blood. In the blood it went to the RBCs, and it burst open the RBCs, and it led to the release of hemozoin, which gives the fever and chill. At the same time also helps in the maturation of the gametes. Now fertilization will not happen in man, so that gametes will be collected by the mosquito, and that gametes are going to undergo fertilization, and that will lead to the formation of zygote. That zygote will form the sporozoids. Mature zygote will form sporozoids. The sporozoids will be stored in the saliva of your mosquito. So remember, students, your sporozoids will look something like this. Okay, let's say this is the salivary gland. Okay, the sporozoids will look something like this. Okay, the sporozoids will be there, which will be stored in the salivary gland, and they will be waiting to infect the next person. Clear or not? All of you, can we go back to NCERT and see if we missed something? All of you in the chat box tell me, yes, clear, okay, what is happening? Clear, okay, Pakka? Yes, very good, okay. So students, here let's go to your NCERT and we will see about your malaria. Okay, this chapter is so big, but if you see, the chapter is a good chapter, right? It makes a lot of sense, and it's important for us to understand that these diseases exist. Okay, look here, all of you. I'm going to reduce the size a little. See, when the mosquito bites another human, sporozoites are injected with the bite. Now, this is a cycle, okay? So, what has happened? Mosquito has bit a person, okay? Mosquito has bit a person. Now, you know that the sporozoites, that is a parasite or the sporozoites, reach the liver cells through the blood. So, through the blood, the sporozoites have reached where? Up to the liver, okay? In the liver, what is going to happen, student? The parasite will reproduce asexually. This is very important. I will write it there, okay? One is going to happen asexually, one is going to happen sexually. So, here, the parasites reproduce asexually in the liver cells, bursting the cells and releasing into the blood. Understood? So, they will continuously reproduce asexually, burst open the liver cells and go into the blood, okay? Now, the parasite reproduces asexually in the red blood cells, bursting the red blood cells and causing the cycles of fever and other symptoms. Released parasite infect new red blood cells, okay? So, here also burst opening happened, that is going to affect new cells. Now, sexual stages, that is gametocytes, develop in the red blood cells, but remember that gamete formation will happen in the RBC, but fusion will not happen, okay? So, here sexual stage where the gametocytes, male and female gametocytes will be developing in the blood cells. Now, say some other mosquito comes and bites us, they are going to collect the gametocytes with the blood meal, okay? Now, they injected Sony. With this, when they inject Sony, they would have taken some blood and gone from Sony, you know, in that blood, let's not say anything, but yeah, let's say the gametes are there, okay? Now, what is going to happen? That gametocytes, okay, will get fused. That is, the female mosquito takes up the gametocytes with the blood meal. Fertilization and development takes place in the mosquito's gut. That's why I told you, fertilization, all the process is happening in the gut, but storage is happening in the salivary gland. Clear? So, fertilization and development takes place in the mosquito's gut, and then it will be stored in the salivary gland. And what happens? Another time the mosquito goes and bite another person, from the salivary gland the sporozoites will enter. So, this is a cycle. Clear to all of you? Any doubts? If you understood, lot of fire emojis in the chat. Now, one thing I have to mention here is what? Asexual reproduction is happening everywhere, okay? Asexually it is multiplying, okay? Here also asexually it is multiplying, here also asexually it is multiplying. Clear? Asexually it is multiplying. Okay? Yes? Clear? Both the places asexually, and here what, students? Here they, as mentioned in your NCERT, I will write here one more word, okay? Okay, I'll just write the gametocytes, okay, so you do not get confused, like, ma'am, where does the gametocytes happen? Okay, so here I'm writing gametocytes are taken, okay, taken with the blood meal, okay, blood meal. Clear? From the gametocytes, here what is going to happen? Fertilization is going to happen. Fertilization is going to happen. Clear to all of you? Topic is done. Okay, is there any other topic that you want me to discuss? Okay, topic is done. Common diseases I am not going to do, as I mentioned before, because that you can do easily, and before also I have done videos, so you can check my RNA videos. But main topics I have discussed. After this you will do some questions and then we can all leave. Okay, yes. So, fertilization, students, is going to happen. That is going to lead to the formation of zygote, and that zygote itself will mature to form your sporozoites. Okay, this entire thing is going to happen in the gut of the mosquito, but sporozoites are going to be stored in the salivary gland, waiting for another, person to get bitten. Okay, so it is going to be stored where? In the salivary gland, but here what is going to happen? Fertilization and formation will happen in the gut of the mosquito. Should I write it outside only? Development in the gut. Okay, clear? Done, done, are done. Very sad, I know. Clear? Perfectly okay. Anything did we miss? Again the cycle is going to continue because this salivary gland, this sporozoite, again it is going to be injected to another person with the bite. Okay, inject another person, the mosquito is going to inject another person, and in that other person's body also this is going to continue, right? So this is a cycle, a complete cycle. Since I am talking about mosquito, I think mosquito is biting. Okay. Yes, even me, I am also disappointed. So students, any doubts, please ask me right now. The most malignant one or the fatal one is your Plasmodium malariae. Plasmodium falciparum. Okay, Plasmodium falciparum. Done, done, or done? Malaria is done. Shall we do some questions? Now typhoid also, just one few words are there very important. Other everything is just terminologies which you can do. Okay. Typhoid students, it's caused by Salmonella typhi. Okay. Salmonella typhi. Students, again fever, chills, okay, body pain, nausea, all that is normal in malaria. Okay. Malaria. Clear? So all that is there. The symptoms I am talking about, okay. Salmonella typhi is the causative agent of it, and you have to remember about Typhoid Mary, okay. You have to remember about Typhoid Mary, the person who spread typhoid, okay. Mary Mallon, her name was, she was a cook and she kept spreading, you know, typhoid. So her name is called as Typhoid Mary. Her actual name is Mary Mallon, okay. Mary Mallon, and then she was called the human carrier of the disease, okay, because she went to different houses for cooking, and without washing hands, without, you know, maintaining the regular hygiene, she ended up, you know, killing a lot of people by spreading the typhoid to many other houses, okay. So Salmonella typhi is the causative agent. I mean, yeah, causative agent. Clear to all of you? Any doubts, ask me right now. Don't wait for, you know, Shankranti to come so that you can ask me. Okay. Now students, as you know from the this only you will understand that how it would have spread. It has spread through the, with the help of contaminated, okay, contaminated food and water. Okay, contaminated food and water is the main source of why typhoid got spread. Be it in Mary Mallon's case or anyone's case, same thing is going to happen. And what are the symptoms that you notice? Usually the symptoms are going to be fever and chills, and Mary Mallon also had the symptoms, but she ignored the symptom for, you know, working, and that is why many people had to, you know, finally she was like, you know, she was staying alone for a very long time, and then she passed away. Okay, that is the story, also very disheartening story, right? Like people isolated her because at the starting people did not know what was causing the issue. Later, once they got to know, they isolated her, and after long years of loneliness, she passed away. Okay, so that is what I've read. Okay, now I don't know the exact story because I was not there. So yeah, fever, okay, very severe abdominal pain. Okay, very severe abdominal pain will be there. Again, chills, all that will be there, students. This is normal. Now in fever, it is mentioned in your NCERT, right? How much it is mentioned? 39 degrees Celsius to 40 degrees Celsius. So such questions can come. Okay, if something is mentioned in NCERT, that is sure it is going to come. Now students, one more thing is we can identify with the help of Vidal test, right? Vidal, okay, test is used to identify it, and there are some, you know, vaccines like your TY21 which can be used to, you know, get cured of typhoid. Clear all of you? Yes? This is your entire thing about typhoid. Okay, I've just mentioned few things. Okay, so you can remember. See, again, I'm not going into the details. So, like details, nothing details, only this much is there. The causative agent, how it is caused, symptoms, and then what is the test and what is the vaccine. Got it? Clear? All of you? No doubts, right? Anyone has a doubt? Please let me know right now. I'm asking again and again and again. I don't know how many times I've asked now at this point. So many times I've asked. Yes or no? So many times already I have asked. Now it's your job to tell me that, ma'am, I'm having this problem, I have this doubt. Okay? Yeah, I was not there that time, unfortunately. That's why I'm not very sure. Okay? Otherwise, I would have got to know more. Okay? Yes, this is clear. Students, malarial symptoms also, again, fever, chills will be there, body pain will be there, nausea will be there. Okay? Yeah, that's it. Okay? Same, almost same symptoms. Clear to all of you? So, if you want, here we can go and we will see the symptoms as well. Okay, here amoebiasis before this. Okay, here you can see, okay, the rupture of RBC is associated with the release of a toxic substance called hemozoin. From here only I have told, okay, I have not made it up, it is here. Okay, so here you can see hemozoin, which is responsible for chill and fever recurring 3 to 4 days. When a female Anopheles mosquito bite, the sporozoites will be transferred. Okay, now students, here you can see that common cold is here, disease, again see vivax, malaria, falciparum. In that, falciparum is the most fatal one. Okay, so here you can see plasmodium falciparum is the most serious one and can be fatal. This is the question that has come. Okay, clear to all of you? Any doubts? Any other doubts? No, right? Okay. Now we will go to questions. We'll do the questions, all of you. Yes or no? Questions? Let's do some questions. Okay, students, I've added snips of NCERT, so don't worry. Okay, I've added snips of all other disease. So when you're taking this notes, make sure that you're reading this. Okay, you're reading as well. Ready? Quiz. One by one questions we have to deal. Okay. Yes, let the energy be high. Questions will be done. After that, we are done for the day. Okay. Okay, ready, steady, go, right? Ready, steady, go. Yes. First question. Which of the following is not a symptom of malaria? Okay, which among this is not a symptom of malaria? First question, let's see the fastest fingers. Which of the following is not a symptom of malaria? High fever, severe headache, muscle pain, dry cough. Okay. Let's sit for 2 minutes. By then all of you quickly tell the answer. Which of the following is not a symptom of malaria? Yes, Jerusha. Faster you should tell the answer. Okay. I can see one answer, but not sure if it is correct. Very good, very good. Okay, first answer was correct answer. Dry cough is not a symptom of malaria, right? Dry cough is not a symptom of malaria. Good job, good job. Soni was the first one to answer and then Madhumati answered. Very, very nice. Okay, very, very nice. Clear? Okay, good job. Next question. Which of the following is not a mode of transmission of HIV? Okay, which of the following is not a mode of transmission? Okay, blood transfusion, sexual contact, sharing the needle, mosquito bite. I specifically spoke about this today, so I think all of you should be in a position to answer. I think, Newton, you're not supposed to think, you have to answer. No thinking. DJ Tililulu is here. Huh? Yes, very good. Because ma'am, you mentioned today that mosquito bite is not going to cause HIV because of the fact that mosquito will digest their blood meal after some time, and HIV is usually, how do you know about virus? If there is a host, they are active. So mosquito cannot be the host. So since they do not have a host, they are going to completely, you know, like they are going to degenerate and they are not going to cause, or when the mosquito goes and bite another person, the HIV is not going to get transported, okay? But that's not the case in our Plasmodium vivax and other things, right? So this is very, very important, okay? Mosquito bite. Next question. Malignant tertiary malaria is caused by Plasmodium. So here, do not worry, okay? Yes. So here it is mentioning malignant, solely focus on that word, and caused by which Plasmodium, okay? By which Plasmodium? Yes. So you people tell, no, Dililulu is the Solululu, like that, Tililulu, his name is Tililulu. Okay, yes, go for it. One by one, all of you can answer. Okay, I can see here fast. Very nice, very nice. The answer is going to be falciparum, right? Falciparum, malaria, Plasmodium falciparum is the most malignant one, okay? Madhumitya, Jarusha and Divya answered first, okay? First, very good. Now, next question. Interferons are secreted by. Interferons are secreted by, this I taught you in the first class, okay? And students, all those notes will be available on Vedantu. Those who are joining late will not know the channel name. Vedantu NEET English, okay? And biology by Gopika. These two Telegram channels, all the notes will be available. Do you want to miss such beautiful notes? Okay, if you don't want to miss, quickly join the Telegram channel. As you can see, the notes. See, look at the notes. Look at the notes. Look at the notes. Okay, all of you can see how beautifully the notes is written. So if you do not want to miss the notes, see, look at the notes. So make sure that you join and take the notes, okay? Yes, answer this. Very good. Virus infected cells. So this is the same technique that we are using for your immunotherapy, okay? Immunotherapy, right, in cancer, okay? Cancer treatment, we are using the same principle of using alpha interferons. Again, virus infected cells are going to release this, so to alert the other cells that I have been infected by virus, you please don't get infected. So here the same theory we are going to use for immunotherapy, okay? Clear? Next question. What are the protein codes and the genetic material present in HIV? That means, what is the genetic material present in HIV? Okay, genetic material present in HIV, you have to tell. Ah, very nice. Okay, tell me, what is the genetic material present in HIV? A for Ammu ma'am, pakka. Yes, I can see, I think most of you got it very rightly, or everyone got it rightly. It is going to be single-stranded RNA, right? Single-stranded RNA is going to be there. Now, shall I ask you a question about this? Tell me this also. With that only, tell this also. What is this I have drawn? Those who are telling lollipop, I will kick you out of the class, right? So, yes. Punishment, severe punishment will be given for those people who are going to tell this lollipop. Okay? Tell the answer for this. What is this? What have I drawn? It is related to HIV, that's why I have drawn it with this question. Okay? Like this, many structures will be there. Yes. What is it? Philo, check the spelling. No, what is GP? I want GP. Full form of GP. Gopika production. Very good, Newton. Very good. Glycoprotein. Why are you people telling globular protein? Okay. Glycoprotein. Glycoprotein, not globular protein. Okay, glycoprotein. And in this, now the next question. Label this for me. 1 and label 2. 1 and 2. 1 and 2 quickly in the chat. What is 1? What is 2? Type 1 and 2, then only I'll understand. Because you people are already telling GP 120. But before understanding, okay? Yeah, they are with actin. They are with actin only, like actin globular protein. Yes, very good, okay? So students, don't make confusion with globular protein and glycoprotein. Glycoprotein is your proteins plus your oligosaccharides. So don't confuse and do all khichdi of everything, okay? One is a part of your immune system, one is somewhere else, okay? So don't get it confused. So first one is going to be GP 120, glycoprotein 120, and this is going to be GP 41, right? 41. Clear? So who is going to bind with the CD4 receptor? 1 or 2 is going to bind with the CD4 receptor in the charts quickly. Tell me who is going to attach to the CD4 receptor. Again, globular proteins also, they are there in the immune system, immunoglobulins, that way. Okay, not directly immunoglobulins. Okay, tell me who is going to be. Very good, ma'am. It is going to be 1, right? 1 is going to go and bind with the CD4 receptor. Okay, CD4 receptor. Very good. Next question. Yes, next question. Colostrum secreted from the mammary gland contains which type of antibodies? Colostrum secreted by, which type of antibodies? Tell me the answer. Last topic questions. Last topic I told you, no, I did not put the question, so I'll be putting this time. So yeah, here is your question. Yes. Very good, IgA is going to be rich in your colostrum, right? Rich in your colostrum. Very good. Okay. How many of you got full marks so far? The immune deficiency becomes marked once HIV starts destroying macrophages, T helper cells, B lymphocytes, natural killer cells. All of you tell me how many of you got full marks? Yes, I can see Sony's message, Divya. Yes, tell me the answer. T helper cells. Okay, T helper cells, and very good. Jerusha, Sukanya, Sony, Madhumati has gotten full marks. Okay, as of now they are raising the hands. Okay, so yes, T helper cells, when the T helper cell starts destroying, that's when we get to know that we are going to a chronic stage, and a small fungal infection, a small bacterial infection is becoming full-fledged infection. Now students, in Tattva, two things have been given which I wanted to inform to all of you, but I forgot. Okay, that is infection from mycobacterium. Okay, they had mentioned that infection from mycobacterium, that is your TB, no, mycobacterium, that is tuberculosis. Usually you would see in AIDS patient, TB will become very high. Okay, like in some cases we see jaundice becomes very high, no, same way here also your TB becomes very high, and because of which that person can go to a fatal situation. So that is again because of the increase of mycobacterium. Okay, and same way you have toxoplasma also increase will happen, and this can cause, you know, become fatal in the case of AIDS patient. Okay, I just wanted to inform mycobacterium. Okay, now next question. The cells of the malignant tumor exhibit what? Okay, exhibit what? The cells of malignant tumor exhibit what? Okay, metastasis, contact inhibition, high differentiation, slow proliferation. Okay, see, other things which are there in the other cancer cells don't tell. Especially in malignant, there is one thing which I mentioned to all of you. So tell me that. Okay, tell me that. Yes, very good, very good. Okay, perfect, perfect. All of you are doing a great job. The answer is going to be metastasis. That means where they are going to continuously spread from one place to another place, they will keep spreading. Okay, next is going to be your homework question. So all of you can read the option here, but put the answers in the comment. Okay, homework question in the comments. Read, I am reading the question once to all of you, don't put the answer. The secretion of antibodies by B lymphocytes provides passive immunity, humoral immunity, cell mediated immunity, innate immunity. Okay, this is your homework question. I want to see all of your answers in the comments, and also put your marks in the chat box. Okay, put your marks in the chat box. Now students, clear to all of you? Any doubts? No doubts, I hope. Okay, answer should be in the chat box. Now this is the video you are checking out. All of you can go down, first of all like the video, and students, here your still offers are still available, but let's also check the price once again. Okay, we will just open it here and view the course plan, and yes, so the price has gone back to normal. That is why I wanted to tell you people, the price has gone back to normal because of, I told you it was a Diwali sale, and the Diwali sale is right now over. Okay, but what I want to show all of you is that all this is there, whichever you want to take you can take, but it is about the NEET treasure box. Okay, NEET treasure box, all yours. See, now as soon as you finish human health, see, as I told you before also, now you have finished human health. Okay, what you're supposed to do, you have to go here and find chapter wise PYQ and search here, where is human health and diseases. Okay, see, look here. Okay, here, see, human health and diseases is here. So when I continue in this lane, I will be able to get the PYQ test. Click on the PYQ test. Okay, view results. Now that is going somewhere else. Okay, my performance, last time's performance is here. Okay, I attempted only 2 questions, so here performance is going to come. Clear? So I want all of you to attempt all the questions given here. Be it 10 questions, I want all of you to attempt your human health and questions, because it is very important for you, very, very important for you to know how much you have done. See, now you practice some PYQs with me, but that's not enough. Okay, you are a NEET aspirant, you need to practice more, double, triple, you have to practice. And I want all of your comments in the chat box, the answer for when B cells are released, it comes under which type of immunity? Is it humoral, cell mediated, or passive immunity? Just let me know in the comments, and also let me know how the session was. All those notes will be available to you within few minutes, so all of you can take maximum, you know, benefit of the notes as well. So thank you so much for joining. I hope all of you enjoyed the session. See you all soon, and the timetable also will be coming soon. I will definitely post the short where the timetable is going to be there, which all of you can use, where I will be also dedicating some time for your coaching. Okay, so thank you so much for joining. I hope all of you enjoyed. See you all next time. Tata, bye bye.